Hello everyone, I'm Mike Sjaastad. I'm with DeNovo Software and we welcome you today to our joint webinar with the Broad Institute. We're going to be talking about the use of CellProfiler for image analysis and data generation, and then the use of FCS Express for Image Cytometry to analyze that data, visualize it, and generate reports. Today we have Dr. Mark Bray from the Broad Institute, a CellProfiler guru, and he's going to be going through a couple examples of image analysis of data sets with CellProfiler. I believe it's going to be a nuclear translocation data set and a colony counting data set. Is that correct? Great. And we have Sean Burke with DeNovo Software, our FCS Express guru, and he's going to be showing analysis and reporting of the results from the CellProfiler analysis of the very same data sets, and give you some examples of how we can set up analysis, presentation, and one-click reporting of your results. So we're very excited to be compatible with CellProfiler. It's the first image analysis software we've been compatible with, and there's quite a few users out there. So just to orient everybody about how this works, I'm going to show you one quick slide before we get started. And this slide is a slide of the general workflow. And as you can see, we think of workflow as being broken down into these two major steps: your image data generation steps, and then your analysis or FCS Express step. So in the blue box, you'll see there's three smaller steps. You do your image acquisition, which is typically on any imaging system. CellProfiler is compatible with so many imaging formats. It really can be just about any imaging system that's out there. And then within CellProfiler, in this example, you would set up your image segmentation, your thresholding, and your analysis, where you would generate objects and information about those objects. So that's feature extraction. And at that point, you can save out what's usually called a data set or a multi-parametric data set that has the object list and the variables that are measured. And oftentimes you save out segmentation maps and image analysis maps along with you have your original images. At that point, if you've done this in the mode that makes the data compatible with FCS Express, it's a very simple step to open it up and import into FCS Express to do further analysis of the data, generate plots, graphs, and charts, and then set up automated reports. And I won't talk much about that because we're going to have a live example of both of those. So with that, like I said, please feel free to use the question bar in your GoToWebinar tool window to send in questions at any time during the presentation, and we'll address some at the end and some of them live. We're going to go ahead and get started, so I'd like to introduce Dr. Mark Bray.
All right. Thank you. It's good to be here. Okay, I think we're set.
Great.
Hopefully I can see my screen just fine. Okay. Well, thanks for the introduction. It's good to be here to be talking to all of you. Like I said, I'm going to be covering the CellProfiler part of this, and we'll be covering two pipelines today. First, a yeast colony counting assay, then followed by a nuclear translocation assay. I'll be going into detail on the colony counting pipeline and spending a bit less time on the translocation one, but I'll make sure to point out the differences between the two. So as you can see here, this is the CellProfiler Interface. The software is free, as you all probably know, and it's available from cellprofiler.org for Windows, Mac, or if you're really hardcore, you can also get it for Linux as well. In this case, I've preloaded the pipeline, and I've already pointed the default input folder to the location of the images for the colony counting pipeline, and the output folder to where I want the output to go. And in this case, we will be working on two input images. This one here, which shows a dish full of red and white colored colonies. So we'll be analyzing this images for the colonies therein. And then we also have a template image, that's just simply a binary image that we will be using just a little bit later. So with sort of that as an intro, we can start analyzing these images by just clicking the Analyze Images button. And while the pipeline is proceeding, I can actually start explaining how the pipeline works. The very first module is the load images module, which, as the name implies, loads the image, which in this case is 6-1.tiff, and it gives it a name, a rich color. That's how we refer to it there on out in the pipeline. Since most of the image processing and object modules expect a grayscale image, we'll use the color to gray module to split this color image up into the red, green, and blue channels. These individual channels are going to be used for different purposes later in the pipeline. Then, because these images are acquired with uneven lighting across the image, or there's also uneven thickness of the agar that the colonies are seated on, the images also need to be corrected for their intensity prior to any further processing. So to do this, we're going to use 3 modules in succession called Correct Illumination Calculate, and we're going to use one of these modules for each channel of the original image. So now the pipeline is actually completed. I can go and double-click any of these modules and bring the display window up to the front if I choose. Each of these Correct Illumination Calculate modules produce an image called the illumination correction function that basically represents the background intensity across the plate. And then the next module, the Correct Illumination Apply module, will then subtract the illumination correction function from the original image in order to accentuate the colonies therein. So by double-clicking the Correct Illumination Apply module, you should be able to see in a little bit the 3 channels, red, blue, and green, the illumination function in the middle column here, which is kind of a smooth version of the original image, and then the subtraction of the 2 to give you sort of an enhanced version of the original images in which the colonies are a little bit more clear. Once that's done, the image math module is then used to combine the corrected blue and green images into one image by simply adding them together. This combined image is going to be used a little bit later in the pipeline and to subtract the green and blue contributions from the red channel. Afterward, we then retrieve the plate template, the binary image that I showed you at the beginning, using load single image. And then we'll be using this image a little bit later to crop away the edges in the exterior region of the plastic plate. So even though this plate template appears as a binary image, binary meaning it's basically just black and white, the image is still loaded as a grayscale image by load single image. So we then use apply threshold to binarize the image. In other words, to convert this grayscale image into a binary image by simply thresholding it. Now at this point, we're ready to align the images together. Now in this case, we're just using one single image of a single agar plate. But if this was a full-blown experiment and we were using multiple images of various plates, it's most likely that the position of the plate within the image would probably vary from sample to sample, just in the way that we acquire that sort of thing. So we use the align module in order to line up the plate template with the plate image, or the channels from the plate image, in order to account for the experimental variation in the plate placement from image to image. So once we're done with that, we end up with a set of new images in which all of them are registered with respect to one another. We can then use the mask image module to mask away the plate edges in the region outside the plate altogether. So to mask away something means basically to exclude those regions, the plate edges and everything outside, exclude those from consideration, which will be very helpful for object detection. And we do this twice, once for the combined image being the sum of the green and the blue images, and then again for the red image. So we have 2 masked images in this case. Then, at this point, another image math module is used to subtract the combined image from the red channel image in order to get an image that accurately provides the redness of the colony. And I'll show that here. So here's the red plate, the combined plate, and the difference between the two, in which the red colonies are enhanced. So that is the end of the pre-processing portion of the pipeline. Now we use identify primary objects to identify all the yeast colonies, both the white and the red colonies within the plate. We will use the red channel image since both the red and white colonies are bright in this image. So you can see that here. The minimum and maximum diameter, which is one of the settings in this module, should be adjusted to just anticipate the types of or the sizes of the colonies that you will get in the typical image, as well as the smoothing and maximum suppression distances that are also settings in this module, depending on the how well these colonies are being segmented or separated in your images. You can use a few of the image tools to sort of zoom in on each of these panels to get a better sense for what the results are. In the display window, you can see the raw image over here to the left and the resultant colony segmentation over here on the right. Again, zoom in a little bit just to kind of get a better sense. Each of the colonies that have been found are color-coded. The colors are actually arbitrary. They're just there as kind of a visual aid, so you can see how well the colonies have been identified and then separated from its neighbors. So that is, we've now pre-processed the images. We've now done the object detection. We have our colonies now. So now we can actually start the actual measurement portion. In this case, we're just using two measurement modules, measure object intensity and measure object size shape. And for that, for the intensity measurement, we're just simply specifying the colonies as our objects, and we're measuring the intensity of the colonies against the subtracted red image. That basically gives us a sense of the redness of each colony. And then we can also measure the colony morphology by using measure object size shape, and that makes morphological measurements of various kinds, such as perimeter, area, etc. So now we've completed all the measurements that we need to do. And then finally we come to the few modules that are specific for creating and saving output that's appropriate for FCS Express. First of all, the convert objects to image. This module takes an object of choice, in this case the colonies, and takes the labels that were assigned by identify primary objects and converts the whole thing to grayscale by turning it into a 16-bit set of integers. Once that's done, you can then take that converted image and save it to the hard drive. All these images, all these intermediate images that are shown sort of in the background are not actually saved to disk until you use a save images to do it. So in this case, we're going to be taking this label matrix, this converted image, that we just simply called colonies, giving this a name, colonies label, and we're saving it as a 16-bit TIFF. And that is being placed in the default output folder. And lastly, export to spreadsheet is then used to save the per image and per object data as comma delimited files, or CSVs for short. The important point here is that FCS Express expects a certain file name nomenclature for it to work properly for these measurement files. So for the per image file, we've chosen to call it image.cptoc. And for the per object colony data, we've called it colonies.cpout. So since I've already run this set of data, if I then switch to the output folder, we can see that the colony label image has been saved. And we also have the colony cpout and the image cpot, which contains all the measurements that we need. So that finishes off the colony counting example. And at this point, I'll switch to the nuclear translocation assay. Now, this pipeline is made for images that were obtained from a 96-well plate. And in this case, for each well, there are two channels. There's a DNA channel for the nucleus, of course, and a GFP channel for the translocating protein. So I can actually show you examples here. So that's of the GFP. And scrolling down a bit. That's of the DNA. So in this case, because it's a 96-well plate, there are of course 96-well images total, so we're not actually going to be analyzing the full image set. Instead, we're going to be using something called test mode, which is a really great feature for allowing to preview the results of a pipeline. To access it, just go to the main toolbar, select test, and then select start test run. So at this point, several new icons appear which allow you to step forwards and backwards through each module, and then also pause execution at a particular module. So for now, I'm going to start running the pipeline for the first image set. And as the pipeline progresses, you'll actually see the slider sort of start from the top and start progressing down to the bottom until the end. So in load images, we're going to be using that in order to retrieve the images as we did before. But in this case, we're actually going to be taking an extra step. We're also going to be extracting metadata from the file name. So here we have load images. We're extracting the DNA channel, which has channel 2 as the part of the name, and the protein from channel 1. And here we have a couple of extra things that you may not have seen before. This kind of complicated looking expression over here, what it basically does is grab the row and column identifiers from the file name and stores this per image information. So for example, in this file name here, channel 1, 89H05, the row information would be the H, and then the column information would be the 05. Both of those bits of information are essentially grabbed and saved as part of the per image info. We'll come back to that in a little bit later. In this pipeline, there's really no pre-processing involved. We're going to go straight into the object identification in identify primary objects. There's a little bit of adjustment of the typical diameter of the objects, the thresholding method, and the types of declumping that's used in this case. And you can see that it does a pretty good job distinguishing the nuclei. Again, I'm going to zoom in a little bit just so you can take a look. So these things just need to be adjusted in a way that's appropriate for your particular assay, whether it's colonies or nuclei or something else. Then we use the identify secondary object module in order to use the nuclei objects as input, as well as the cytoplasm image also as input. And then we use these nuclei objects and grow outward using the GFP image as a guide in order to obtain the cell objects. And so the output is shown here. Again, we'll zoom in a little bit just so you can get a better view of the output. And you can see how with every nuclear object there's a cell object surrounding it that's also enclosing it as well. So these are the two objects that we're going to obtain for this particular assay. So with these two objects, we can now start our measurements as before. Measure object size shape, again, is used to come up with the morphological measurements from the nuclei and the cells. And the measure object intensity module to obtain the intensity of both nuclei and cells from both the cytoplasm image and the DNA image. So those are the measurements that we're obtaining for in this case. Again, we use convert objects to image to convert the nuclei objects into a nuclei grayscale image. Each nuclei gets its own unique number for the nuclei and for the cells. And then we save both of these out as images. And in this case, we're going to use a kind of a special feature of save images to create a unique file name for each image based on the metadata that we obtain from load images. You just simply specify the image that you want to save, in this case cells, a single name as the method of constructing file names, and then you type in whatever prefix or whatever text you want to be part of the file name. And then what you can do is by clicking in this edit box here and then right-clicking, you can actually insert the metadata that you want to become part of the name. And I've already done that here, and that's why these two labels are shown in green. That just indicates that there's metadata there. So as it goes through, if we were on channel 1, 89H05, the H would be substituted in the row tag here, and the 05 would be substituted in the column tag here. This image will be saved again as a 16-bit TIFF in the default output folder. and that is for the cells and, again, for the nuclei. We're using metadata substitution in both in order to get a unique image name out. And then, as before, we use export to spreadsheet to save the per image data. Here, again, we label it image.cptoc, and the per object data for both objects, nuclei and the cells, as nuclei.cpout and cells.cpout. And just as just in case you're wondering, the reason why there's a yellow bar here and kind of a warning flag over here is that it just indicates that in test mode, it's not actually going to write out the spreadsheet since we're only analyzing one file at a time in test mode. But I've actually run this pipeline a little bit earlier on the full image set, so I can show you what the output looks like. You can see here that cells.cput, nuclei.cpout, image.cptoc, those files have all been output. The cell label, the converted label matrix is still of the cells is here, in addition to that of the nuclei. And you'll also notice that, as I mentioned before, the row and the column information was inserted into the file name as we specified in load images. So that is basically it for the kind of the walkthrough of the CellProfiler pipeline. If you go to our website, you can find more examples that are similar to this, along with some example images. And even if your questions are not answered during this webinar, you can always go to our online forum and ask any of your image assay questions there. And we'll certainly be happy to answer them. So at this point, I'll turn it over to Sean to talk about the details about how to use these files in FCS Express.
And this is Mike. I'm going to step in just now. It's a good time while we're changing over. If you have any CellProfiler specific questions, go ahead and type them in, and we may have Mark answer them live if it's a general enough question. Otherwise, we'll address it by sending a message back to you.
And hi, this is Sean Burke. We did have two questions from Frederick Henry regarding CellProfiler that maybe you could address, Mark, real quickly.
Okay.
The first was, can the align module be used with images taken part of as part of a time series to correct for slight drift?
Yes, it could. The way to do that is a little bit roundabout, but briefly, what the align module does is that it can take any number of a set of images and align them all. However, the images will always be aligned to the first one in the group. So for a time series, a time course, what we usually do is pick one image out of your set. Let's say you have a movie or your time course split up as an image sequence. Oftentimes we'll usually pick the first one kind of arbitrarily, just we need to register it against something. So we'll pick the first image as the input for the align module. Let me switch over to it here. So yes, this would be the first image out of the series would be the first image. And then you just simply specify the other images that you want to align for the second, third, and however many more. You can add as many images as you want if there are, say, multiple channels, for example. And you have the option of aligning them all the same. That's what similarly means. Or do a separate sort of alignment for each one. That's what separately means. The one catch here is that if you want to use the same image as this first one, you would probably need to load it using load single image as I did here. So you'll have a load images that will load in all the files for your time course, and then a load single image that will load just that first file, basically as this pipeline did here. But yes, it should definitely be possible. And as far as alignment goes, it uses various image features to align with. So the kind of underlying assumption is that your images are certainly different. It is a time course, but they should be similar enough that alignment in terms of features should be at least somewhat possible. So that's the short answer.
All right. And with the module steps that you performed in CellProfiler, are they appropriate for ZStack images taken on a confocal microscope? Would that be possible?
Yes, that would also be possible. The main difference would be changing load images in order to accept a ZStack. So here, the very first setting is individual images, simply completely separate, distinct individual image frames. You also have other options here. SDK movies, and I say movies as if it were a time course, but it could also be a ZStack as well. SDKs are, I believe, a metamorph format for what's simply just a type of TIFF. AVIs, MOVs are also supported, and also a TIFF and Flex and ZVI. ZVIs are, I believe, Zeiss movies are also supported as well. The basic options are the same. Load images doesn't change much by selecting these, but you also have a few other ways of sort of grouping movies together. And one nice thing is that you can run through several sets of movies or sets of stacks, and CellProfiler will sort of keep them, will keep track of them individually. It'll certainly record measurements from each slice, but it will also put in a marker indicating that these measurements come from one stack, these other measurements come from the second stack, and so on and so forth. But aside from that, the rest of the pipeline probably wouldn't change that much. So the short answer is yes, it can handle them. There's usually not too much tweaking that has to be done, but yeah, it's the sort of thing that you just give it a try and yeah, check it out, see what happens.
Great. And maybe we could ask one more question before we move on, and then we'll save the rest to the end. We had a question for the nuclear translocation assay. How do you make sure that cell labels correspond to nuclear labels?
Okay. One convenient aspect of the Identify Secondary Objects module is that there is always only one cell per nucleus. So you're pretty much guaranteed a one-to-one ratio. When you output them in the spreadsheet, you'll see that if you were to, say, output the nuclear measurements and the cell measurements in one file, that's something you would do if you were just working with the measurements in using CellProfiler Analyst or one of the other ones, not for FCS Express, but you actually have the option of combining objects into one file rather than splitting them up. If you're combining primary objects, the nuclei, and secondary objects, the cells, they would all be placed on one row, and you would know that there's a one-to-one correspondence between the two. It's just one of the features of how Identify Secondary works. Now, if you did had another identified primary objects and identified another set of objects entirely, then you couldn't guarantee that sort of correspondence. There's not necessarily a one-to-one correspondence between two sets of primary objects, but with secondary primary objects, there will be.
Great. And we also had a few questions about using the metadata extraction or the extraction feature there. So one thing, we'll come back to this at the end if there's still questions about it. But as part of working with the CellProfiler team, FCS Express has put together some, or DeNovo Software, we've put together some tutorials for generating these pipelines in CellProfiler. One of the things that we added is a more detailed description and some links to websites that show you how to do the metadata regular expression extraction. So we come back. Yeah. Go ahead, Mark.
Well, we also have help available next to the metadata extraction line here, which goes into at least some amount of detail on how to actually write these expressions that grab metadata. Granted, it is a little bit complicated, but I think once you get the idea for how it's structured, it should be fairly simple.
Great. So let's leave a few of these questions to the end, and maybe we could transfer over to me at this point, Mark, and we'll save the last 5 or 10 minutes of the hour for questions about CellProfiler and FCS Express. So it looks like I have control here now. So for a lot of you, FCS Express is probably going to be a new piece of software for you unless you've been in the flow cytometry world at all. But FCS Express has been developed as a powerful flow cytometry data analysis and presentation software, and we just released our new Image Cytometry product. And CellProfiler was one of the first pieces of software that we became compatible with and that we're able to show data for. There's a lot more in the works, but CellProfiler has worked very well so far. But the main principles of flow cytometry and image cytometry, such as taking multi-channel fluorescence data and plotting the data in scatter plots and histograms, so you can look for statistically significant differences and changes. They're the same between imaging and flow. So we can now take that image cytometry data a step further by leveraging the tools that we already have in our flow cytometry software to make analyzing your data from CellProfiler much easier. You're going to be able to use the original images to confirm your results, and we make reporting this data and getting publication quality figures and collaborating with your colleagues very easy. So right now, what you're seeing on your screen is FCS Express, and let me open just a little bit of a different layout here to start with. What you'll notice first about FCS Express is that we have a very similar look and appearance to Microsoft Office products. You can see here that we have a ribbon, and we have all the tabs that you're probably used to seeing, or some of the tabs that you're used to seeing in PowerPoint or Microsoft Word or Excel. And the whole idea is that if you know how to use Microsoft PowerPoint, you should be able to come into FCS Express with whatever data you have and be able to instantly get up and going with your analysis. So just like in Microsoft PowerPoint, you're going to have this blank screen in the background when you open up a new layout, and we'll show you that. So you're going to be presented with this, and on this blank screen is where you're going to put your plots and your pictures, and everything that you see on here can immediately be exported to Microsoft PowerPoint very easy by doing the save layout as a PowerPoint presentation. But we can also go out to PDFs, we can export the page as a picture, and many different types of saving type of modules there. But what I want to show you first is some CellProfiler data. This is the images, or two of the images, that came from the nuclear translocation assay that you saw. I'm going to start with just two images so that you can get a feel for the software and how it works and the power of it. And then I'm going to go into the nuclear translocation data set and also the colony counting data set that Mark had showed earlier. So the whole idea in FCS Express is that you can bring your data in to something called a data list, and you can see here that these are the files that were put out by CellProfiler. We have a cellular image for, say, a 5 and 10 minute time point, and a nuclear image for a 5 and 10 minute time point. And what we've done is we just inserted a few plots, and we can do that through our Insert tab. And you can see that just like PowerPoint, you can insert text boxes and shapes and pictures, but we also have things like histograms, proliferation, bar plots, dot plots, color dot plots, picture plots. So just like in PowerPoint, we can say click on a dot plot, draw a place that we can put a dot plot, grab this data from our data list, and now you can see all the different parameters that were exported from CellProfiler. In this case, we chose to export everything, so it's a very long list. But we can choose what we want for our X and Y parameters. So maybe what we're interested in is our area shape on the X parameter and our integrated intensity of the cytoplasm on the Y. And when we do this, you can see we get a dot plot that represents all the cells that are in the cells05.cpout image. All right? And this is exactly the same as, or very similar to this plot that we've already inserted. The main difference is we formatted some things about this plot. This is another great feature of FCS Express, is that you can format anything about the plots and the displays that you're seeing. So you can see that we made the dots a little bit bigger. We made some changes to the title here. And you'll also notice that some of the dots are colored. We have black dots, green dots, and red dots. And let me delete this plot, and we'll focus on this now. And what these dots represent is something that we call gating in flow cytometry. So a gate is kind of a threshold or a region of interest. It's that kind of equivalent to the imaging world. And you'll notice that on our top plot, we call this a picture plot, we've defined some new regions or gates. And what we've done is we've colored these green and we've colored these red, and you can see that we have something called a gate view over here that's showing you the names of these. And what this means is when I actually move this gate to another section of the image, you can watch down here on the dot plot that the cells that are highlighted in green change, because the cells that are highlighted in green fall within this gate. So we can really zoom in on any part of the picture that we want and see where those cells fall as in reference to some other parameter within the data. So we can also set up as many gates as we like. In this instance, we've set up 2 different gates here. We can overlap these gates, watch where the cells overlap. And all the cells that appear in black are just cells that don't fall within this gate. So this is a quick and easy way to come in and zoom in on your data and get some real population-based analysis from dot plots and see where those cells fall on the actual image themselves. So we can go from gating picture plots and showing that on dot plots, but we can also go from dot plots with a gate on it, like you see here, and display those cells on the image. So you'll notice that as I move this blue gate, the cells that are highlighted in blue on the image update in real time, and this is going to allow you to really zoom in on, say, an outlier and figure out where that outlier is and maybe what's different between that outlier and everything else within the data set. So we can also apply these gates, or we can use these gates to narrow in on a population. So if we only want to look at the cells within the green gate on this dot plot, we can actually just drag this gate onto the dot plot. And now you'll see that we're only viewing the cells that fall within that gate. And we can again do that with our red gate if we want to look at just them. And we also have something on the side here that we call a cell gallery or a data grid. And this is showing us whatever parameters that we want to look at in the data. In this case, we just chose area shape. We could choose any of the many parameters that come out here and any combination of them. But you'll notice that we also have a cutout of these cells in the data grid. What this means is if I want to zoom in on, say, maybe these 3 or 4 outlier cells here and see what they look like individually, I can drag this over to the data grid, and you can see exactly what those cells look like because the data grid is now gated on this dot plot square gate. So these are just some of the basic gating features that we've come to know and love in the flow cytometry world, but that we can do in the imaging world now as well. And we had a question about how do we do our gates, or how do we make our gates. Anytime we want to insert a gate, we can go to our gating tab. We have a few different types of gates to choose from, which should include any shape that you could ever imagine. So if I wanted to say make a polygon gate, and we can just click on the dot plot a few times. We can call this my new polygon gate. You can see that we've created a gate. We've also created this gate, as you'll notice, as a hierarchy. So of this new polygon gate, it's only showing the cells that fall within this picture plot gate. So we've said, look at the cells within this red gate, plot them on the dot plot, and then tell me what the percentage is through this new gate of those cells that fall within this dot plot. All right? We can make these hierarchical if we want, or anytime we can actually pull this gate out, and we can make it so it's not a hierarchical gate, and now you'll see that our statistic updated because of that as well. We can draw these gates directly on the image, as I mentioned. So if we want to draw a rectangle gate on our DNA image, we can just draw that there. And now we're getting the percentage of the cells that fall within this gate. And for this gate, we've defaulted not to show the cells that fall within the gate in color, just to make it a little bit more visually stimulating. So you can see how a gate and the gate statistics are very, very powerful and easy to use. And I'm going to come back to some of these questions. They may be answered as we go along in the data sets. I want to make sure we leave enough time to cover the CellProfiler pipelines that Mark outlined for us. So here's just a general idea of how gating works, inserting plots, and some of the more simple things that we can do. But Mark has generated, or let's see, let me bring up my next page here, the nuclear translocation assay. So CellProfiler has put out the data from this nuclear translocation assay, just like Mark had should. We have our cells.cp out, and this cells.cp out encompasses, this one file encompasses everything that was included in the entire plate's worth of data. So all 96 wells, there was 2 images per well. And you'll notice that in FCS Express, we've inserted something called a heat map. Heat maps can be used to display any size of plate-based data. You'll also notice that on the y-axis here, we can choose any of the parameters to display in the heat map that we've defined in CellProfiler. All right, and what this also means is that we can define gates or thresholds or regions on this heat map as well. So if I wanted to say create a gate, a well gate, I can actually click on a well in the image, and now we can use this gate to only look at the cells that fall, or the images that fall within this well. I can also select multiple wells at the same time. I can draw a box to select them, and we can select non-contiguous wells as well if you'd like. So in this manner, what we've done in this assay is set up, you'll notice here in my edit gate section, we've set up a whole bunch of column gates. So we've said column 3, 4, 5 through 11, and you'll see when I choose to show that gate that we're actually treating these 4 wells as replicates, and we're pooling this data so that we can look at all the images from these 4 replicates at the same time. All right, and again, we've done that for all the columns within this plate. So what this means is that we have some negative and positive controls set over here on the plate, is that we can set a gate on those just like we did here, our positive controls, and we've pooled all that data. And we can go to, say, another page within the layout that we've chosen. And we could say that on this plate, or in this dot plot here on the top, we're going to show all the cells. So this is everything that's from all the images within the heat map. And then we're only going to show the cells that fall within the positive control wells and the cells that fall within the negative control wells. And what this is going to allow us to do is define objective gating populations for our experiment, right? So you can see here that we have an example image from our translocated cells and an example image from our non-translocated cells. And we can come on the dot plots and say, here's our translocated population, and we're doing this by looking at the cytoplasmic staining and the cell edge staining. So as there's less cell edge staining, that means that there's more staining within the nucleus. And we've also looked at, again, the non-translocated cells, and when we define our gates in this way, it's a completely objective manner, and it's a very, it's a much more, I guess, scientific way of doing things rather than kind of looking at the image and saying, I think these are translocated because it looks like it's going to the nucleus. We can really come in and define that objectively at this point. So, again, here is another page where we've actually come into the heat map data, and we've said, let's look at some picture plots for that. And we've gated all of these images and all of these plots on something here called our single well gate. And this is going to allow us to change this gate, look at the titration series, and have everything update in real time. And you'll notice that here we've put in a picture plot that doesn't have a background, and we're showing the cells that are non-translocated in blue and the cells that have translocated in red, and what that image actually looks like in real color or grayscale. So this is a great way to come in and review the assay, make sure it worked. In this case, we can see that we've gone from about half and half from translocated to non-translocated, and as we go down the rows or the columns in the assay, they're becoming more and more translocated. So this is great, but what we really want to do in the end is report our results and have a nice publication quality report. What we've done to do that is we've inserted a different dot plot for each of the different columns or the pooled wells of data in the assay. And we've looked at what the percent translocated is for each of those populations here. And in our next page, we've actually defined, and let me change our orientation here, we've actually defined a regression curve based on this data. And what this means is that each of these points is coming from a specific column worth of data, and they all link to this translocated cells gate. So if I decide that my translocated cells, I need a little bit more leeway for that gate, I can adjust it. And our regression analysis is going to update in real time with that gating change, right? So this means that you don't have to export or use a bunch of data in Excel, create a plot, and then if you need to change something about that data, you have to go back and recreate that plot. This is all being done and calculated in real time within the software. And just so you can know, there's also, we can look at all this data in histograms. In this case, we chose to display each of the different columns in a kind of stacked histogram here. We have our positive control in blue, our negative control in red, and all the different titration concentrations going along within that. And again, all these update in real time to gating changes, and that's one of the main advantages of using FCS Express. And in the end, when we're ready to maybe publish this report, maybe we want to put it out to PowerPoint, all we have to do is go to Save Layout, Save as PowerPoint, and let's give this a name, and I'll call it Delete Me so I remember to delete it. And we'll open the presentation after saving and just this page. So you can see now PowerPoint automatically opened up, and we actually have a PowerPoint presentation of this data. All these are just single PowerPoint objects now that we can move around. We can send this to our colleagues to work with, and it's a great way of getting your data from just raw data to results very quickly. All right, so that's kind of the nuclear translocation data set. You've seen some of the things involved with that, and I'm just going to take a few minutes here to go over the colony counting data set that Mark had put together. You can see here that we've actually brought in the raw data for this colony counting assay. You can see the red colonies and the white colonies. We've also defined just a few small gates on this colony counting image as well. And we've inserted a third picture plot where we've put in a black background. And this is going to allow us to kind of see the colonies a little bit better. One thing that we can do in FCS Express is if we want to zoom in on this plot at any time or this image, I can right-click and choose Magnify Axis. If I wanted to see what these colonies actually look like, you can see that we can do that very easily. We also have unlimited undo within FCS Express, so if we want to go back a step, like to unmagnify the axis, we can choose to do that. And you'll notice that we've also put in some dot plots here for this data. And Mark had said that we've put out a few different parameters. One of them is going to be the mean intensity of the red, and one is going to be the mean intensity of the red, green, blue together. So if we only look at cells or colonies that fall to the right of this, we can assume that they're red. Colonies that fall to the left of this, we can assume that they're white. And we also have a colony area parameter. So we can easily define large colonies, medium colonies, small colonies. We can put in as many gates as we want at any time if we want to have further subdivisions of this. And you'll notice that we also have a little statistics table here that we've put in. Now this is considered a gate statistics table. I can do the same thing here if I right-click on this plot and I choose gate statistics. you'll notice that we come up with a table, and this table can be moved anywhere you want within the layout. If we right-click on it, we can choose to show any of the statistics involved with these gates. So let's choose to not show the file name because we're not really interested. And this is a great way that you can just generate quick statistics for any of your data. And you'll notice here that we have 2,860 colonies on the plate, 146 are red, 2,700 are white. We've also subdivided these into large, medium, and small. And we've also put in our Big Colony 1 and Big Colony 2 gates, which are actually these gates that we've set up on the colony counting plate here. All right? And anytime, if we want to zoom in again on any of these large colonies, we can maybe, or let's look at the small colonies. It'll be a little bit more obvious. We can now see that here's all the small colonies. Here's a picture. If we want to look at the red colonies, we can drag that gate on there, pull out only the most red colonies out of everything. And the great thing about FCS Express also is that we have the ability to batch process. So what will happen is you don't usually take a picture of one colony counting plate. You take a picture of 100 colony counting plates or even 1,000 for a huge experiment. What we can do is add them into our data list here. So if you've taken 100 pictures and you've generated 100 colony.cpout files, we can actually go to our batch processing tab, and we can add some actions here. And we can say, if we want to save these to PowerPoint, we can click that and put that in there. We're going to get an error because I didn't give it a file name, but we'll just keep going on with that. And if you want to export all this data to Excel as well, we can just do this export to column mode. And again, I didn't enter a file name, so I'm just going to continue there. But any of this data you want to bring to Excel, all we have to do is drag and drop it to our Excel column mode report, and then that data parameter is going to be stored to the batch process. And when you run the batch process on, say, 100 samples, all of these statistics are going to go out to Microsoft Excel at that point. So very quick and easy ways to do batch processing and deal with hundreds and hundreds of files. All right. And as a quick summary, we're coming to the end here before we open up for questions. CellProfiler output was the first format that was compatible with FCS Express, and it's completely supported. We offer many tutorials and example pipelines so you can get up and running with your CellProfiler data very quickly. We leverage some of the most powerful tools of flow cytometry analysis, and you can apply all those to your image cytometry analysis. This means that you can move from population-based analysis to single-cell level just at the click of a button and by dragging gates around. It's very easy to do. You can have full interactivity between your analysis and all the data plots in your analysis. And we're the only software package or data analysis visualization software offering a free reader. So if somebody or one of your colleagues does not have FCS Express, the full version, they can download the reader version. What that's going to allow them to do is you can send your file to them, and just like Adobe Acrobat, where you can send somebody a PDF, they can open it and read it. There's certain things they're not going to be able to do with it, but they can get an idea of what's going on with your analysis, and they can move the gates and all of that. CellProfiler and DeNovo Software, we both provide many resources for our users, like the tutorials, the sample data, and online forums and excellent support. So that's going to allow you to come to either CellProfiler for that part of the analysis or to DeNovo Software for your final part of the analysis to ask questions and get answers to them and get off and running with your analysis very quickly. CellProfiler is free, of course, and it's available at their website, cellprofiler.org. FCS Express and DeNovo Software, we offer a free 30-day trial at our website. So all you have to do is visit the website, go to the download tab, and download FCS Express Image, and you should be able to get off and running with your CellProfiler analysis very quickly. Again, at the end, if you have any questions that you remember after the webinar or that you come up with, you can always email us at support@denovosoftware.com. So at this point, I'd like to open up the floor to questions. I don't know if, Mike, you wanted to address some of these questions or present them to Mark and I?
Sure, I can read through some of them. Mark, we have you and Sean. Everyone's live on the phone here. Let's see, going back up to, we answered the gate question.
I see a question that just came in here on the bottom. It asks if FCS Express can do t-tests and other stats to compare populations, along with doing density and bar plots. These are all things that we can do. We actually have a way of doing statistical analysis. You can do any type of mathematical analysis that you want through the use of what we call tokens. Tokens are bits of statistical information that come from your plots. So at any time, if I wanted to say, choose this number of events and stick it out here, that's going to appear as a token. Let me make sure I put this in the correct spot. And you'll notice here that these tokens can come from any of the statistics. Actually, that one doesn't make a lot of sense. But the idea here is that with tokens, we can also create custom tokens or formulas like you can in Excel. And when you do this, we have the ability to use any type of function that you really can imagine mathematically. You can see here that the list is actually quite exhaustive. But these can all be used to do your analysis and get statistical comparisons. We can also insert bar plots and scatter plots. You saw the scatter plots in the regression analysis, but we can do the same thing with bar plots as well.
Okay, great. And I see here in the list of questions a general trend towards curiosity about other image formats that we're compatible with. And I just want to address that by saying we maintain information on our website as we announce formally image format compatibility. Most of you are familiar with CellProfiler. A lot of our registrants today came through the use of CellProfiler. So you know that hundreds of image formats can be read into CellProfiler, so that's quite a gateway to bringing data into FCS Express. But I wanted to let everyone know we have active collaborations both in the research imaging software markets and the high content markets, and hope by the end of the year to have quite a few formats easily available, if not sooner. So just keep an eye on the website. We have your email addresses, and we'll be making announcements as that comes out. But lastly, we have announced our own image cytometry format. We can provide documentation on that. If you're working with software and you're able to write out in that format, we will always be able to import that standardized format. And it's a format we're putting out in order to make it easier to work cross-platform. So there was one other question here for Mark. It says, can you go over the load images module again for the translocation example? How is the metadata extracted? Is that something you can address
yes.
quickly?
Let's see. Is that something I should take back control of the screen so people can see?
Let me send that over to you, Mark.
Okay, hopefully that's visible to everybody. So, let's see, in a little bit more detail, basically to extract metadata, you have the option, an option which is normally which is down here in Load Images, extract metadata from where. And you have a choice between doing it from the file name, so if the information you want to obtain is in the file name, you can check that. Also the path, sometimes the folder name itself can be taken as metadata, or you can do both. And you'll have boxes for each one, or both, depending. What this essentially is in this edit box here is what's known as a regular expression. Basically, it's a type of sort of language, as it were, that illustrates, that allows you to grab text of one type or another. Let's see, I can try to show very briefly what this indicates here. Regular expressions can be a little bit hard to understand at times, so what we've done here is given you a regular expression editor, and what it shows here is, at the top is the regular expression itself. The second box is just some test text, which is hopefully which should be the first file name in your input folder. And what this shows here is channel 2 dash. It's looking, so what the expression is basically looking for these characters in this order. After the dash, you have in brackets 0 through 9 with a 2 in braces next to it. That means look for a number in which there are 2 of them, one right after the other, followed by a dash here. In this case, in the test text, you do see there's channel 2 dash and then 2 numbers. The next is the more complicated part in which the question mark P and then a name in between the greater than less than signs. This is called a metadata token. It's just a name for what the metadata is, or whatever you want to call it. What follows after that is the actual text you want to capture or way to describe that text. So this is saying look for the letters A through H, and if those exist, grab those and give it the name row. And then follow that by look for a dash afterward this here. So the metadata tag that's shown in red is also color-coded in the text just so you can see at a glance that you're capturing the right thing. After the dash, there's another question mark P with column in the greater than less than signs, and so we're capturing another bit of metadata, and we're giving the name column. And then after that, this next bit of text just indicates look for the digits 0 through 9, any one of those digits, and there are 2 of them, which is why the 2 is in braces, and give that the name column. So after, in the test text, you see an A, a dash, and a 0, 1. It's going to grab both of those, the 0 and the 1, and it's going to give that name column, or assign that the name column. And then after that, there's a dot and a tiff, and that just kind of sort of closes it out and makes sure it lets it know that this is where to stop. So that's just sort of a simple example of how this works. Like I said, regular expressions is a whole kind of language unto itself, and so that's why the help by this setting is actually fairly extensive and actually links to an external website, so you can read up on it if you want to do more complicated metadata extraction.
Great. Okay, we have one last question, and then I'll wrap up. And it's for Mark. It says, can CellProfiler with FCS Express calculate the median and MAD per object populations after classification for use to calculate the SSMD stats of the positive versus negative phenotypes?
Okay. Well, so for the intensity measurements, measure object intensity, for any of these modules, you can hit the question mark and get a box illustrating not just how the module works, but also the available measurements that you can get. So you'll see here that all the available measurements, integrated intensity that Sean was showing, mean intensity, you can get standard deviation, the min, the max, integrated intensity on an edge, and a few others. You mentioned the MAD, I believe, and in the current release of CellProfiler, it's not there, but we've included it in our source code since then, and so for the next release, the MAD will also be in there as well. And these, just as the module name indicates, these are on a per object basis. So all of those will be exported for each object found to the final CDOut spreadsheet, and FCS Express will be able to work with those.
Okay, great. Mark, maybe you can change over to me as the presenter really quickly, and we'll wrap up.
Okay.
So for those of you that are still with us, one of the great things about today is we're working with open source software, and on the FCS with CellProfiler, and on the FCS Express side, there is a free 30-day download if you want to try the Image Cytometry version. I believe you can now see the CellProfiler homepage. I just wanted to make sure you know that where to go to take a look at this software. You can download the software. We had questions about the images used today. At the bottom of the homepage, you can go to the image benchmark collection, and you can go to image sets. If you want to download some of those images, it's what we used in testing the software. For FCS Express, And both companies have a great reputation for supporting their products. We do an awful lot of support in both cases, actually, through the website. On the DeNovo Software homepage, if you want to learn more about the Image Cytometry or the Flow software, you can see there's immediately available on the homepage a 10-minute video highlighting the features of both. There's access to the upcoming webinars, so today's webinars, and importantly, all of the recorded webinars, which can be a great resource towards learning new features. Today's event will be compiled and put up on the website within a week, and access to downloading the 30-day demo. So you can find application examples and tutorials about using CellProfiler and other examples in the Image Cytometry software on the web page. You do not have to download and install the software in order to have access to those. There's a page dedicated to Image Cytometry and a number of example applications where we've actually got CellProfiler pipelines and examples set up for you to take a look at. So I wanted to thank everyone for attending today. It was a great turnout, a great audience, and we'll stay in touch. Thanks again, and thanks to Mark and thanks to Sean for their presentations.